Good afternoon, everybody. I'm going to speak about one of the research works that we have been doing here, which is related to model-based systems biology. And we have applied object process methodology to visualize molecular biology processes and systems. So just a few words about model-based systems biology. A few citations from relatively recent papers. You can see a transition is occurring in biology from the molecular level to the system level that promises to revolutionize our understanding of complex biological regulatory systems. Said by Kitano in 2008, formal models promise to change the face of biology and medicine. Abstraction and high-level reasoning will likely play important roles. And the third one, at this opportune time, conceptual biology is being born. Okay, I would like to spend a couple of minutes just to show and remind you about object process methodology, OPM, principles and symbols. In OPM, the system is represented in a single type of diagram, which is called object process diagram. And there are two main entities: object, which can have one or more states, and the process which transforms the object. It can generate an object or consume an object or change the state of an object. So each process has a pre-process object set, which is the precondition, and post-process object set, the post-condition. And the precondition is checked before the process execution. So if all the objects are there and if there is a requirement for them to be in certain states, if all those conditions are met, then the process executes and does what it has to do, and the outputs that appear in the model. So for example, in biology, enzymatic reaction is a process, and in order for it to happen, there has to be an enzyme which acts as an instrument. This is the white lollipop circle symbol here, and the substrate which is consumed by this enzymatic reaction process, and the output, the result, is the product. And in parallel to the graphical representation, there is a text output that is generated automatically in OPL, which is Object Process Language, simply a subset of English that is understood by anybody who knows basic English. We also developed OPCAT, which is the software environment for modeling systems with OPM, and it is downloadable freely from the website of our lab, which appears also in the site of Visionaire. One of the ideas in OPM is the idea of in-zooming or refinement of processes and also objects. So if we look at the left, there is a gene expression which consists of mRNA life cycle as one of the processes, and if we zoom into it, we see mRNA life cycle here with all the details. So this is the same process as here, but now with many sub-processes and intermediate objects. So this is a basic idea that we take advantage of. So again, to summarize, OPM is a bimodal representation. One is a graphical, so a set of object process diagrams which look like this, and corresponding automatically generated sentences in OPL, which is a subset of English. So for example, here we have an object called robot with two states, disengaged and engaged, and there is a process of engaging which takes the robot in its disengaged state and changes it to engaged. And this is exactly what is shown in the automatically generated text here. Robot can be disengaged or engaged, and engaging changes robot from disengaged to engaged. So OPM unifies in a single model the function, structure, and behavior of the system. It can model hardware, software, regulations, humans in the loop. It has a B-model expression through graphics and text, as I said. And now, more specifically, let us digress a little bit to talk about molecular biology. We start with the macro level of whole organisms and organs, and they are made of specializing cells. Each cell has a size of about 1 micrometer, and within it are thousands and hundreds of thousands of various molecular interactions, biological, biomolecular interactions, pathways like the glycolysis that is presented here. And each one of them is a biochemical reaction that is represented as an example here. And we want to be able to model the system at the various levels of detail. So this is a link to the book. It talks about object process methodology, and this is a screenshot of our website, which I'm not going to go into now, but you are welcome to visit it and download not only the software, but also a lot of papers that were written in the lab. There is also an ISO standard 19450, which is in the process of being ratified. We have a meeting in Frankfurt, Germany, in a couple of months to submit it for final voting. And this is going to be the official definition of OPM to be used by industry and anybody who wants to have a standard conceptual modeling language. This is a paper that was published about 2.5 months ago in a very prestigious journal called the PLOS One Public Library of Science by Judith Somek and Mordechai Hoder and myself. Judith is a PhD candidate who is now finishing and has submitting her research. Mordechai Hoder is a professor in the Faculty of Medicine, and I'm from the Faculty of Industrial Engineering and Management. And this is just the first page, but you're welcome to look at the paper. It's freely available because PLOS One is a open access journal, which means that anybody can access it through the web with no payment or no difficulty. Okay, this is the real thing. As you can see, all kinds of. So this actually what the talk I'm giving now is, of course, related to the paper. It's a brief overview of what we did here, or a subset of what we did in this work. So what do we do in model-based systems biology? There are lots of research papers and experimental findings that are written there. This goes into a computational model, usually in systems biology. We use OPM, as I said, and this is an example of an OPM model of the decay of the mRNA dissociation model. This computational model represents all the experimental knowledge that is embedded in free text articles that are published, all of them currently online, of course. We have the tools to execute the model. This is to say we actually run the model. So here you see the same screen, but the colors indicate what process is currently running. So the big process that is running is this blue, light blue one, and within it this dark blue one is the one that is currently executing. Green means objects that exist, and brown is the state at which each object is. Okay? So whereas here you see a generic model, here it is an actual execution of the model in runtime, just as you would run a computer program. So in stage one, we verify that the computer simulations are compatible with the experimental results that are in the paper. And if not, we fiddle with the model until we get the behavior that reflects the experimental results. This is a cycle that keeps on going on and on. In the next stage, we can add new biological conjectures, things that have not yet been found or verified in experimental setting, but mechanisms that might be operating behind the scenes. So we augment the model with them, and then we execute the model again, and this either verifies the results or is a reason to do some more wet lab experiments, which are usually very costly, and we can spare and save many of them by doing these so-called in silico experiments. This is just an excerpt or a screenshot of a model that is based on 22 scientific articles that were read very meticulously, and each fact was incorporated into the model. This is a relatively high-level view. You see mRNA life cycle, which consists of two sub-processes, decay and nuclear import and transcription, and you can see all the objects at high level that are involved in it. It doesn't mean a lot to most of us who are not biologists, but biologists know very well each object in each process here. And on the left you can see this is the top level, this is the dark blue, the system diagram, or one level down. But as you can see, there are many levels of nesting, that something like 10 or 11 levels of nesting at the maximum, which is really a lot. This is another example from the decay and the nucleus import, which is the first sub-process, is zoomed into, and you can see that now this is blue. So this is just one level down, but you can see how many levels more there are. This is another one, deadenylation, which is one of the sub-processes here, okay, the third one. What appears here in gray is a symbol of something that is not quite certain, because it is not, it is a conjecture. So DHH1 to CCR0 binding is currently a conjecture that has not been verified, and should be and can be verified by carrying out an actual wet lab experiment, which can take a long time and cost quite a lot of money. Like I said, there are many levels of nesting. This is one of the deepest, level 10. You can see 1.1.1, 10 times deep, but still all you can see here are objects with states and processes, just as we saw at the top level and at the level below it, which means that regardless of the level of detail, we still talk in terms of objects and processes, and this is one of the strengths of OPM, that it is very generic and very simple on one end, but allows you to go into any level of detail, and all the diagrams are self-similar, almost like fractals, if you're familiar with this. Another important issue is that we can detect knowledge gaps, which we define as lack of knowledge regarding an object or a process in the system. And in the course of this work, we found 22 knowledge gaps as a result of executing the model. And we even found two papers, or two sets of papers, that contradict each other. And both of them are published, and somebody can find one paper and take it as a fact that it goes this way, and the other paper says it exactly the other way around. Another thing is the experiments establish facts, but it's one thing to find outcomes from experiment, and another is to be able to explain the underlying mechanism that can explain why those facts were found. So what we did is we augmented the model with a conjecture and iteratively improved the model, which includes now the conjecture, to fit the laboratory findings until it was satisfactory. And then it still remains to be checked in the lab that this conjecture also meets other conditions that are expressed in the model. So we did new in silico experiments before doing them in the lab and predicted system behavior under varying conditions by executing the model. Again, it remains to be checked in the lab. I want to spend a few minutes on another issue that I mentioned. Some of you may have heard it in the COG-InfoCom conference in Kosice a couple of months ago. The human spatiotemporal comfort zone is a zone where people feel comfortable with respect to size and time orders of magnitude. So it's the zone in which humans can comfortably view and imagine things existing and happening in space and time. We cannot, because our senses are not geared to dealing with living cells, let alone molecules, we have to draw much enlarged models of them. Okay. Also, some processes take such a small amount of time, microseconds or even less, that again we cannot comprehend them unless we spread the time orders of magnitude more in order to be able to track what is happening. So the idea, and this is how it is linked to Visionaire, we want to be able to have a mechanism or an instrument that takes molecular level interactions that are very small and happen very quickly, and to control both the size and the time that it takes for them to happen. So we want to translate the system in action into the human spatial temporal comfort zone and combine inspection-driven with model-driven visualizations. These are the two principles that underline the next generation of covering. Covering is like a cave, but for realizing nature, a cave for realizing nature. So here is, in more numeric terms, the cognitive limitation we are talking about space and time. So humans are about 1 meter order of magnitude in size, and they can think about time of about 1 hour. These are the golden, let's say, the neutral metrics that in space and time that people can easily relate to. So the tolerance is minus, from what I found, is -5 to +6 orders of magnitude of 1 meter, which means it's between 0.1 millimeter to 1,000 kilometers. This is something that humans can relate to with their senses. It's like something like a hair, the thickness of the hair and the distance between two countries or two very distant cities. With respect to time, if we take 1 hour as the middle, again, we can think about relatively easily -5 to +6 orders of magnitude. So again, 0.1 second to about 100 years, so which is the, you know, the order of magnitude of age of human beings. So, and also there is Miller's magic number, which is the concurrent number of items which people can hold in their short-term memory. It's 7 plus or minus 2, with 5 and 9 being the lower and upper bounds. And like I said, molecules are much smaller and the reactions are faster, so we need to transform them to the comfort zone. So COVERN is an acronym for Computer-Based Augmented Virtual Environment for Realizing Nature, and it's an instrument for doing next-generation research in systems biology. It's based on cave technology and creates a totally immersive living cell environment. It opens up a host of exciting new possibilities in biological research. So this is how I envision it. There are already caves that can show things like this, but the idea is to have the models not just static, but being driven by an OPM model that's such as the one that I showed you before. This is one mode of operating the system. Another mode would be just inspect what is going on in real time or near real time in a living cell and project it in this instrument. Here is another, again, rendering that I made up from, you know, I used the pictures from the cave in Aachen, but superimposed, you can see some molecules inside them by extending them. So the inspection-driven mode is to visualize a visualization that originates from inspecting the cell in action, and the model-driven mode is driving this by running in the background the OPM model, the dynamic OPM model of how the system behaves over time. Okay, so to summarize, we've seen that in OPM we use stateful objects and processes, and this is a sufficient set of entities to model complex systems. We focused on biology, but as you may know, we have been doing it in virtually any domain and at any level of complexity, as I've shown, with 10 or 11 levels of depth of nesting in the biological model. And not only can it be used for engineering, but also for science, because science and engineering are both about systems, and OPM is capable of conceptual modeling of systems in general. With science, we want to understand existing systems, and in engineering, we want to design and build new systems. I mentioned that OPM is becoming the first ISO standard, 19450, for conceptual modeling, and this is an opportunity that I hope some of you will adopt as a universal modeling language for the purpose of modeling the systems that you're engaged in.